Primeiramente, professor, em nome da pós-graduação do Instituto de Biofísica e em nome do próprio Instituto e da UFRJ, para a gente é prazer ter o senhor hoje conosco. Today is an honor to have Professor Sérgio Schenkman in this symposium. Thank you very much for being with us today. For us, it's a privilege to have a talk in your level. And in the name of the Biophysics Institute from the Federal University of the Rio de Janeiro and from our postgraduate program, thank you very much to be with us today. Professor Vanderlei.
Okay. Thanks, Susana.
Podem ligar a gravação, por favor?
First of all, I would like to thank Sérgio to accept our invitation to participate of this web symposium. This symposium was organized in view of the pandemic, that we cannot have the people here. That would be our first option, to invite colleagues to come to the Biophysics Institute and give the talks. But that is the way that we can work during this time. I would like to say that tomorrow we'll have the conference of Dr. Guadalupe from Mexico that works with Entamoeba histolytica. And that we have a mix of parasitology, cell biology, and microscopy talks. So that is the way we could organize this meeting. I would like to ask my colleague Narcisa Cunha Silva to introduce Sérgio Schenkman for those that does not hold very well his work, because we have here, Sérgio, an audience of people in cell biology. Of course, all people work with the cruzi. I know you very well, but we have students and you have people from different parts of the world that are in the YouTube system. Please, Narcisa, go on.
Yes, thank you. Dr. Sérgio Schenkman was born in São Paulo, graduated in biological sciences and made his PhD in biochemistry by the University of São Paulo, USP. He was a postdoctoral fellow at the Institut Pasteur in Paris and the MBL in Heidelberg, Germany. He became assistant professor at the Universidade Federal de São Paulo, UNIFESP, in 1984, and afterwards joined the New York University Medical Center in 1988 at Victor and Ruth Nusswein Lab, where he started the very important studies that led to the identification and characterization of the enzyme transialidase neuraminidase of Trypanosoma cruzi. Later on, he identified the mucins as the acceptor of the transferred stearic acid in trypomastigotes. These studies paved the way to many others until now. In 1997, he became full professor of microbiology at the same university, and at the microbiology, immunology, and parasitology department, he already supervised more than 30 post-graduation theses, 11 postdocs, and has published more than 163 papers. He's currently member of the editorial board of several important journals and full member of the Brazilian Academy of Sciences. Since the early 2000s, together with Maria Carolina Elias, Júlia Chagas da Cunha, Sheila Nardelli, Nilmar Moretti, that was his PhD students or postdocs at different moments, Sérgio started important studies on the chromatin, mainly histones, and nuclear organization of Trypanosoma cruzi. The cell cycle progression of epimastigotes has also been his focus, with important morphological and molecular markers of interphases and mitosis being defined. In the last decades, Sérgio published important data concerning the signaling control of metacyclogenesis by TCK2, a kinase that phosphorylates eukaryotic initiation factor 2 alpha, and that is inhibited by him. And at that point, at that time, nutrition, signaling, differentiation, and oxidative stress control come across. So today we will hear Sérgio talking about all these interests together. Please, Sérgio, it's our pleasure to hear you.
Okay. Thank you, Narcisa, for the kind introduction, and Vanderlei. Also, it's a pleasure to participate in this web symposium. It's nice to be part of this team that is giving lectures to you, and I hope my lecture will be exciting and
surely will.
I'm happy to answer the questions. And I choose this team is a recent work that we did. This is mainly produced by PhD students in the lab, and we have the collaboration with a group in Ohio State University, Bradford McGuire, that came to us and with the cyclphilin, and then we studied the cyclphilin in epimastigotes initially, and now we decide to work on the cyclphilin in the cruzi, and I think this is came across our team of working because we are interested also to this oxidative stress in the parasite growth. So I'm going to talk about the Chagas disease. Chagas disease is caused by this Trypanosoma cruzi protozoa. For those that are not so familiar, I'm going to be quick, but mention that the transmission of the Chagas is mainly by oral. I believe it is by oral through these hematophagous insects. And what's important about Chagas disease, there are about 8 million people that are infected in Latin America. And more recently, there are numerous cases in US, Canada, and many other countries due to immigration and transmission by blood transfusion. So Chagas disease is still a problem for us and is spreading. So there are some concerns about the disease that justify initially the infected people, the infected individuals get this acute phase, and then this moves to a chronic phase. And 1/3 of the patients infected with the parasites can develop a kind of cardiac insufficiency, and it's not in this slide, but also they can develop intestinal problems. And from these 1/3, at the end, 30%, some of them can go to do heart transplantation. And you see today in Brazil, 30% of the heart transplants are due to Chagas infection. So what makes me go to the story that I'm going to tell you today is the fact that the cruzi growth depends on the presence of reactive oxygen species. And this has been nicely reviewed by the group of Claudia Paiva and Boza, Marcelo Boza, and they found that these reactive oxygen species they have a role in the Chagas disease. So in the infection, the oxidative environment can favor the parasite growth, while antioxidant can decrease the parasite burden. They have shown this in vivo and also in vitro. The group of Claudia and Marcelo, but many other groups have shown that cells, when they are in the presence of oxidative stress, they support the better growth of the parasite. And they did the experiment using either fibroblasts or even cardiomyocytes to mimic the the heart infection, when they add hydrogen peroxide as a source of oxidative stress, they have more parasites as the control. And the same thing happen when they add antioxidants like catalase that destroy hydrogen peroxide, and this complex is the PEG, that's the polymer that allows the enzyme to get inside the cell, so they decrease the burden of parasite growth in vitro in cells. So it seems clear by now that some level of oxidative stress is necessary to drive the growth of parasites inside mammalian cells, and this is important also during the infection. So the questions we try to answer in our work is to understand how these reactive oxygen species are increased during the cruzi infection, how the parasite does that. And then we also have a question is if the immune response generated by parasite has a role in the generation of this, and this would affect the disease progression. So what I'm going to talk is this immunophilins is a class of proteins. They are characterized because they have this name immunophilins because they bind immunosuppressors. Immunosuppressors are molecules that prevent the immune response, and then they identify first the immunosuppressors. One of them is cyclosporin A, and this several years ago, the cyclosporin A was found to bind a molecule they call cyclphilin, and cyclphilin is one type of immunophilin, and there are other immunophilins that they don't have capacity to bind cyclosporin, but they also bind other immunosuppressors like the FK506, and these are the FKB binding proteins. There are several of these proteins in our cells and also in many other organisms, and this group are called immunophilins. What the immunophilins does, in addition, they have an enzymatic activity, and they are able to catalyze the peptidyl-proteolysis transisomerase in the protein chain, or in solution. Proline can adopt two different conformation. One is here, and that we call in trans, and the other is here because it's so this is catalyzed by the PPAs. There's this type of enzymes, and this occurs in the proline, also inside when the proline is in the polypeptide chain. And the consequence of that is that some of these enzymes change the conformation of the proteins and can mediate several cellular processes. One interesting thing is the cyclophilins are many. They are present in different organisms from even in prokaryotes, in bacteria, to mammalian cells. And here are the main cyclophilins of yeast with the names, and these are the mammalian orthologs. They have different size, but they all have a conserved domain that is called CLD, and then the CLD contains the enzymatic activity, and they have a conserved or more or less conserved structure. And these different cyclophilins, they are different because they have different localization signals in the N-terminals or in the C-terminals, and they adopt, at the end they have different size and act at different place in the cells. What's interesting, one of the most abundant cyclophilins in humans is called the cyclophilin A, is a cytosolic cyclophilin. I'm sorry. And this and it acts in different tissues. This is just an example. Oops, it's moving by itself. And this is the structure of the cyclphilin by crystallography. And the cyclophilins are studied in macrophage, T cells, neutrophils, platelets, endothelial cells, smooth muscles, and cardiac fibroblasts. So what is shown is that cyclophilin affect the signaling in macrophage of these pathways. And also is interesting because cyclophilin is, in some cases, secreted. It's involved in chemotaxis, in these different cells, is involved in angiogenesis, is involved in extracellular migration, protein migration. And this is the intracellular roles. And these are what indeed show what the cyclophilins, how the cyclophilins are modulated. They are modulated by LDL, TH2 to TH1 polarization. So ROS induction is known in smooth muscle cells and in cardiac fibroblasts. So what's interesting in this is that cyclophilin has many, many roles. If you go to PubMed and look for cyclophilins, there are hundreds of papers showing the effects of cyclophilin, and one of them is cyclophilin A. In 2010, this review by Sato, Japanese, I think it's Japanese group, they described that the cyclophilin is related to production of ROS in several cardiovascular disease. So it seems that the ROS, intracellular ROS level in vascular smooth cells, induce the secretion of cyclophilin. Cyclophilin binds to a receptor, and this receptor signals activation ERK that increase the levels of ROS, and this ROS is primarily increased by the activation of the NADP oxidase enzyme, in the case of vascular smooth cells in NOX2, and this occurs, for example, when angiotensin 2 binds to the receptor and signals the increasing ROS production, producing this increasing cascade. Another factor that produces ROS is mechanical stress or many other environmental stress. So the cyclophilin seems to mediate, or is described to mediate all this process. So because of that, several diseases, and I would like to call your attention for the role of the cyclophilin. It involves the myocardial ischemia and reperfusion, inflammatory heart disease, and even hypertrophic. And the cyclophilin is always involved in that. The arrows shows where the levels of cyclophilin affects heart, and this is reviewed by Sizer. If you want to get more information, I can send you later this. So how the cyclophilin acts, how the cyclophilin is involved in ROS production. There is one paper by the group of Bradford Burke that published some years ago, that a cyclophilin A is required for angiotensin 2 in activation. So P47 is a subunity that is represented by the circles. It binds to the cyclophilin. They show this binding, and this goes through the cytoskeleton to the sites where the NOx is activated. So what they propose in this work is that the cyclophilin helps through its chaperone or through its enzymatic action to carry P47 to the activation, to the sites where the NOx is activated, and this occurs after angiotensin activation. So this signal is important. It's not probably they suggest, but it was not proven, that depends on the phosphorylation of the P47 by the binding of the angiotensin. But what basically this work show is that if they knock out cyclphilin in the cells, there's no activation of NOx in these vascular smooth cells. Going back to cruzi, it's interesting that cruzi has 15 cyclphilin isoforms. This is a published paper by Mariana Potenza in June, and they review, at that time they have a limited amount of data from the genome, but this is amazing what we found today with the most recent analysis. There are several cyclophilins from 19 to 110, and these ones contain the CLD domain. They have chromosomal localization. So we studied the cyclophilin 19 because it's the most abundant of the cyclophilin in the cruzi. And then, although there is one work that's homologo of cyclophilin D, is involved in the ROS production. So it's not only cyclophilin. This was the work by the Argentinian group of Jacqueline Bois that they published in 2017. They show that the cyclophilin is somehow related to the oxidative stress damage of the parasite. Just a parenthesis, cyclophilin D is expressed in the mitochondria. So just a quick review of role of the cyclophilin A in disease. There are some publications showing the platelet thrombosis, thromboinflammation, myocardial inflammation in myocarditis. There is a lung inflammation, oxidative stress in aneurysm, in aortic aneurysm, also by the group of Bradford, Burke in Nature Medicine. This is more recent. And then we go to the comparison of the cruzi cyclophilin 19, that is the most abundant, with the most abundant cyclophilin of our cells, of human cells. And then it's amazing, these two proteins are very similar. They are 70% similar. The main difference is related to the presence of the N-terminals only in the cruzi cyclophilin. And what is interesting is if you look out the CYP19 in Brucei, cruzi, and other Trypanosoma, they all have these extra N-terminals, and this is unique for the trypanosomes. We don't know the function of this N-terminals. It has something to do with the stability, localization. Nobody really knows that. So we initially checked whether the cyclophilin was expressed in the cruzi, and then this is different stage of the parasites, and this is a Ponceau gel, this is a BIP has internal control. You see, these are the levels of cyclophilin in the epis, metacyclics, TCTs. They are the parasites that circulate, eclode from the mammalian cells and circulate in the blood. And this is the intracellular form of the parasite. Is just the quantitative analysis showing that it's almost expressed equally and probably a little more in the epimastigotes. What we found that's interesting, that the cruzi cyclophilin is expressed by tripomastigotes of this parasite when they transform into mastigotes. So what we did is one experiment there, we incubate the parasites in normal conditions, and we look of the amount of cyclophilin in the supernate, and there's a little bit of secretion, and this is the endogenous cyclophilin. But then when we measured the amount of mastigotes that forms, very little, so these triangles are the TCT, very little tripomastigotes became this mastigotes. This is a normal situation after incubation at 37 degrees. However, when we add, when incubate the parasites in low glucose or without serum, there's much more secretion, and then the differentiation occurs much faster. This is similar things occurs when we do shock at pH 5, and this is being published by others. It was never found that removal this type of stress induced differentiation, but it seems that the triple mastigotes, they become intracellular mastigotes when something changes, and this may occur during the invasion of the mammalian cells or a certain point. So this tells that the differentiation or the secretion of this protein occurs in the parasite. So the question is this occurs in the intracellular, in the parasites that are growing inside the mammalian cells? And the problem is that I show you that the two proteins are quite similar, so we have to tag the CYP with HA, and to see that, because we couldn't use an antibody against the proteins, because the antibody against the one recognized the other. So we tag the protein with the HA epitope, and this is the slide showing that the tag is expressed. This anti-CYP19 and anti-HA. See, when you use the antibody against the triple mastigotes, when you use the anti-HA, and this is for the M mastigotes, and this is for the HA, showing that we could tag the protein and then is expressed together with the endogenous protein. And this is a gel showing that the HA is in the correct size. And it's interesting because we add the tag, and the expression of the tagged proteins here. So it's not, it's moving too fast. It's not highly expressed. The tag is a little B. And this is the control parasite showing only one band of the CYP, the endogenous CYP is appearing. And this is the quantitative analysis of the two. So the tag is there. So the tag is less expressed than the endogenous protein, and there's no difference in the Y-type parasites. So what, with that antibody, with that tag, we use the anti-HA antibodies to see whether this protein is produced in parasites growing inside the mammalian cells. And you can see here, this is an uninfected cells with anti-CYP19 is against the parasite. It does recognize the cell, but nothing appears in HA. So showing that this is control, this is a DIC, this is the merge image. After 24 hours of the incubation of the parasite cells, you see this is the parasite, this is the nucleus of the mammalian cell. You still see the labeling of CYP19, but it becomes stronger in some points. And then you see that the HA tag parasites are here. So some of the parasites are highly fluorescent and expressing, not all, because the population was not homogeneous. So this is interesting because it serves as a control, interesting control. 48 hours, you see the number of parasites increase, and then you start to see, I don't know if your screen is clear enough, but you start to see some shade, in the cell cytoplasm. But then when we look at the parasites more longer time, we start to see the cell cytoplasm becomes labeled. And we don't see this label in this cell with the parasites without the tag. So this is something due to the presence of CYP. And this is more impressive image at 96 hours. after invasion, you can see, and you can see some of the seep in the edge of the cell, like that. And this is a high mag of that, so we clearly see the extracellular CYP19 and in some positions of the cell. Gregory Pedroso dos Santos, the student that did the confocal analysis, and then you can see the presence, clearly the presence of the HA labeling outside the cells. Then I think this slide is not in the right place. I'm going to go back to that. So what happened with this exogenous CYP? So express this CYP-H, it's not overexpression, it's a little higher expression, and we compare the proliferation of the parasites. And then when we take it 24 hours, you see the invasion and the number of intracellular parasites about the same. But after 48 hours, we start to see a little increase in the amastigotes in the culture. And this becomes more evident at 72 and even more evident at 96 hours when we saw that amount of CYP. So it seems that the presence of CYP, additional CYP, increase the number of mastigotes in the cells. And this GFP control is, for some reason, is a little decreased. And also we did the same, another calculation when we compare the total mastigotes per cell. So this is the mastigotes per cell. We have a number that increases slowly, while the red one corresponds to the parasites expressing CYP-H. We also did an experiment. I'm going to show that later. Then when we express the in the mammalian cells, so we transfect the mammalian cells with the HA CYP, and then the previous slide here, I'm show the expression of the AHA in the L6 cell. So this is GFP, this is the L6HA, and this is just the labeling of with CYP-A. You see HA is now expressed in these L6 cells, and then using this cell line, we see the same thing that we saw with the parasites expressing CYP, more CYP. So when we express the CYP in the cell, we increase the growth of the parasites, and not when you express GFP in the mammalian cells. And you see this is the total number of mastigotes. The same when you calculate by mastigotes per cell. So this result showed that the presence of CYP in the host, in the cell, increased the growth of the parasites mainly after 72 hours, but still the parasites grow. So because it contains CYP, it secretes its endogenous CYP. So to demonstrate that, in fact, CYP19 was important for the growth or not, we generate a CYP knockout by CRISPR using exogenous recombinant Staphylococcus aureus Cas9 complexed with the guide RNA. This cuts the CYP gene in the beginning of the gene, and then we use a donor that recombines using the 5' and the internal portion of the CYP, inserting the blastocidin gene. So this was prepared by PCR. These are synthetic, these are recombinant protein. And then we did this experiment, and we select the parasite using the blastocidin gene. And then we obtained it after some weeks a cell line producing much less CYP. We don't, even after selection, we couldn't get the full knockout, or this could be, I would say, a cross reaction with something like that. We're still working on to be sure exactly how this knockout is. These are blasted resistant. And also we did add back, adding the HA, the same plasmid we used before. So this is the HA plasmid in this condition, and you see that now is re-expressing similar amounts of CYP in this case. Here's the loading control using aldolase. And then I'm going to show this later, it's out of order. So what happens with the knockout replication in the cells? When we look at 24 hours, there's no difference. We couldn't find the parasites. Even at 48 hours, when we start to see something, there was nothing. But then we couldn't see anything more at 96. So this is the KO in the red, a little decrease, and this is the add back. The add back returns up to the same level of original. Remember, I'm going to show again, the overexpressor grows more at 96 hours. Okay, this was out of order. This is also to show the knockouts. I'm sorry, this was, I think the slides move, was not in the right position, but this is to show that most of the trypomastigos, they don't have CYP19, they don't have HA, but they start to express the add-back start to express the HA. So it seems that the knockout was more or less 95%. This is just to show that the L6 cells express HA tag in the cells. And then we took these L6 cells and start to see, okay, if the CYP-A is related to the oxidative stress, as I show you, what happened in these cells expressing the CYP-HA, the parasite CYP-A, using a cell ROS deep red is oxidative stress sensor. So this here is the CYP-A, HA expressing the cells, 3 fields, just to show that the cells increase the ROS. And this, we remove that N-terminal from the protein and it's still expressed. So the N-terminal is not necessary for that. And these are the control cells, L6 cells. There's a little increase in ROS, but it's not significant. We also measured the ROS level of the cells using another probe, CM-H2D-CFDA. And these are the Y-type cells. These are the GFP, and these are the CYP19. So these two methods show that these cells contain more ROS. And then what's important is that when we do these measurements, this is the same results as shown here, but when we incubate the cells with cyclosporine A, that's an inhibitor of cyclosporine, we decrease to about the same levels, the ROS measured by H2D-CFDA, showing that this ROS depends on the presence of something that inhibits cyclphilin. And there is some works showing that the CYP19 is inhibited by cyclosporine A. This has been shown by the group of Bua, Bradford, McGuire, our collaborators. So they show that what we see here is related to the cyclophyllene. Finally, we tried to see if the growth of the parasites is restored using the add backs. Yeah? So we measure the amounts of mastigotes per cell in adding nothing. This is the level in L6 cells, and this is the wild type parasites. When you do the knockout, the level is reduced at 50%. This is significant. We did in replicates, triplicates. When you use the add back, it restores it to the normal levels. But when we add the cyclosporine to all three cases, we decrease to the levels of the knockout, showing that this is 96 hours later, that we are decreasing. Okay, you could ask, maybe cyclosporine is just inhibiting the parasite growth, but then we did the experiment using cat PEG, like the group of Claudia Paiva and Marcelo Boza, and also use SOD PEG, the superoxide dismutase PEG, and we also reduce. And then for the knockout, I'm sorry, the reduction was not so much. Probably there's endogenous cyclphilin, and we also reduce for the add back, showing in fact that the levels of parasites is related to the cyclphilin present in the parasite. But we also test the cells that contains cyclphilin. And it's interesting that these cells support the growth of the knockout. You see the knockout. And they are both inhibited by all these cyclosporine A, CAT, and SOD. And the add back it has the same level of infection, showing that either cyclophilin inside the parasite that is secreted has the cyclophilin already present in the cell, is able to promote the growth of the parasite. So how does cyclophilin 19 does that? We just repeated the experiment of Brad Bertie and in our system, and then we use the cells. We label with CypA, we label with P47 Fox, we label with actin using phalloidin, and then this is a superposition of each case. So CYP and actin appears to have some kind of colocalization. So you see accumulation of CypA, like it was described, in the leading edges of the cells. This is CypA. And these leading edges contain actin, accumulation of actin, because the cells are migrating. More interesting, these images, is that we can see CypA in fibers, in the actin fibers. And the same is true for P47. That also accumulate. So it seems that are the regions that contain the migration of the cells that contain NOx, active NOx. Then we did the same thing for our cells. So these are cells expressing HA. We can see that some cells contain these leading edges here. This is an example. Here is another. But these cells is migrating and it contains these leading edges, and then you can see the filaments, the active filaments. This is better. This is superimposition of these two images. So here's another one. So it seems that CYP19 is working similar to CypA in the cells. We don't know what is the role of the cyclophilin A in leading edge. In fact, this is not discussed, but one I can give you some hint that the cyclphilin are highly expressed in tumor cells, some tumor cells. These are myoblasts. And tumor cells have this capacity to migrate in some conditions. So it might be related to the cell migration capacity or something happening in that that has to do. But NOx is more activated in this case. We also did this by a more sophisticated analysis. We used, because the CPH-HA antibody is quite strong, we could do super resolution microscopy with HA, and then you can see better the accumulation here. And the P47 is just a confocal image because the labeling was much weak. So this is a regular confocal, this is super resolution. We did the merge, and you can see the accumulation of both. We can see regions in the cell that have the accumulation mainly at the edges. This is a high mag of this region, and then clearly see CPA in these trails that probably are microfilaments, and these are the confocal image. So confocal image is not as perfect and super, and you can see better the alignments here. So we don't have a formal proof that CYP19 still don't have. We are looking for, because see there's so much CPA in the cells, but whether this is the mechanism of CYP19 is the same of CPA. Another interesting thing in other images we did in confocal is that in some areas of the cell we have P47 and CPA in stretches of the cell. This is a high mag in the confocal image. You can see the alignment of the proteins probably on the fibers. So probably this is the way P47 is carried through the okay. We did this image in the confocal with the help of Renato Mortara. And these are some shots of that previous movie showing areas that are less localization areas, there's more. So in summary of my talk, we found that the CPH is released by the parasites and it induces the increasing ROS. And we explained that by the capacity of the CPA to transport this P47 subunity of NOx to the edges of the cell and activate the ROS. And then we are proposing that the CYP19 is one of the factors, and we don't think it's the only factor, but that increase the ROS and promote the parasite growth during the infection, and explain why the parasites can grow, or why this CPA is important. So this is a model that we draw that the parasites invade the cells. Inside the pH, the vacuole gets an acidic pH. This induces transformation into an acidote. We don't know exactly when. And then the parasites to secrete CPA. There's triangles here. And this CPA helps to induce NOx. NOx improves the ROS, and then this increases the parasite proliferation. Finally, I'd like to acknowledge this work was mainly done by Gregory Pedroso dos Santos, who is a FAPESP student, PhD student. He's going to finish his thesis soon. Fernanda Midori Abukawa prepared the L6 cells with the help of Greg. Renata Finicio, I didn't show the experiments. Renata was Academy of Science fellow that came here for summer, did experiments of immune response of CPA. Nilmar Moretti was our collaborator now as a postdoc when he started. He's now a professor here at the department. Nimar is the one that started this project and that track continued. Mariana was our task force to prepare the cells and plasmids and knockouts. And then we have the collaboration, have the collaboration with Bradford from the Ohio State, and BJ is a postdoc in Bradford lab. He sent us some signals. And this is what I have today. I'm happy to take questions. I hope you have enjoyed. Thank you.
Yes, Sérgio, very beautiful presentation. Thank you. We have some questions came from this Meet chat, and some of them came from YouTube. I will try to alternate. A question from Professor Norton Hayes. He's amazed that all CP19 are secreted from the parasite to the supernatant in the absence of glucose in fetal calf serum. Is that CP19 in the cytosol? You did show that overexpression of HA TCCP19 was lower when compared to the total of endogenous CP19 present, but the overexpression of the HA CP19 was able to promote growth of intracellular mastigotes. Could you comment a little?
Well, about the secretion, we still don't know the mechanism of secretion. I can go back to the slide here. Let me see. Where is the secretion slide? Here. Okay. So when we compare the amount of CP in the cells, it seems that the CP expression, it disappears. We don't know the mechanism. And initially, we use this system minus glucose and minus FPS because when basically the story is when you add the trypanosomes concentrated in medium, the medium became yellow. And then and they only secrete cyclphilin when the medium became a little yellow. So it seems that the lack of glucose and cetos bovine serum stress the cells and the media become a little acid. So we don't know the absence of glucose is just a consequence of stress, or is because it doesn't have glucose, it changes the metabolism and becomes more acidic. This I try to see if even people don't know why the parasites acidify so much. It just release acetate and things like that. If it does not use glucose, and the trypanosomal metabolism is doing that. Or if the secretion is a consequence of a stress independently of the metabolism. We don't know, to answer your question. And then what we know is possibly when you transform the parasites, he likes to release piece of it. It's like autophagic, but not a real autophagic. He just throw away his protein from the cytosol. This is my impression that is happening. In the acidic pH, this occurs. So how a triple become a mastigote, you start to run and release pieces of membranes and also cytosol, and this, whether this is selective. The other question is whether this occurs inside the cell in the same way as the mastigotes are growing. We really don't know. I think I think others have shown secretion of proteins that are intracellular, cytosolic, but we have, I didn't show you, but we did the Western blots of whole cell cytosol. We found the CP19 there too. So I think it could be like a mechanism of ejecting pieces of cytoplasm. and I think could be related to the fact that many of the protein, the cruzi develop a way to turn over rapidly the proteins when it's changing, mainly when it's changing, instead of doing the proteasome degradation. They use the proteasome for the cytoskeleton because it cannot degrade the cytoskeleton, but they don't do the proteostasis typically. They prefer to release the things out. It's more convenient. And then they start to make things again. They are the same or different according to this condition.
And Sérgio, Vanderlei is asking if CP interferes with the generation time and the cell cycle of intercellular mastigotes, the proliferation of mastigotes. You showed that mastigotes proliferate more, but which is the pace?
We didn't check the cell cycles has a typical way, like counting things. We just see that in 48 hours they double the same, almost the same. This was an important question, whether the intracellular CP was necessary for the division for the cycle. We, for this reason, we add the exogenous CP19 in the cell. I personally think it's hard because epimastigotes, we don't see any difference in growing.
Sérgio,
necessary for a mastigote. I cannot exclude this possibility. It's true.
As you said that mastigotes, let me put a question of myself. Do you know that the intermediate epimastigote forms could have a role in this scenario of CYP19 turning epimastigotes more competent to invade more cells and maintain the infection locally?
In fact, he probably asked trypanosomes invading more cells. And,
But some groups have shown that we have intermediate epimastigote forms.
It could be. I don't know this.
Intermediate between mastigotes and trypanosomes.
Yeah, I don't know if this cyclphilin will be released from. We still have to work on that. Whether the CYP19 is secreted when the parasites burst, or if this is released from the cell like CYP-A, and this could affect further infection or the other things, or this could affect. Probably this could change the immune system. Because CYP, we know that the parasites also bind the receptor of CYP, that is CD147. CD147 is a receptor of CYP-A, and we demonstrate, our collaborate demonstrate that CYP binds. But another question, another thing he asked is whether the intermediate form could be, the change to intermediate could be important. Yes, I think it possibly the increase in secretion could happen not at the beginning, but when the cells start to grow later on, when the intracellular mastigotes start to become this epi-like form, or start to some. I think that the mastigotes, when they are occupy the entire cells, they have a kind of limitation, so they could now start to secrete things and change the shape to differentiate into trypanosomes.
Sérgio, let's turn the talk to some another points. From YouTube, Nelly Damuskis asked if CYP19 could be used to diagnostics, or maybe as a form of following the treatment to see if the parasite charge diminished. Or we have another question related with that. Let me find it here. No, yes, you could please answer that questions about the diagnostics.
Okay, this is a very good question. We asked the same thing, and then we tested 600 patients using the recombinant CYP19, and the question is whether they will produce antibodies against this protein or not. And
Yeah.
it's amazing because I would say that 15% of the patients produce, not more.
Oh,
But we couldn't see any relation with the state of disease because we tested patients at the in the later stages and the early stages or asymptomatics.
but do you think that CP could be involved in the resistance to treatments, parasite resistant to treatment? It's a question of Romário Lopes.
Yeah, so we're trying to see if there's any correlation between that. We couldn't establish a clear correlation by looking about 450 patients, and then we have a second the serum of these people collected 10 years later to see. We haven't tested yet, but my
Yeah.
impression is most of the serum, most of the patients recognize this N terminus. That is different.
Yeah.
We tested these peptides, and all the positive are against that. So and we also, but there is another thing that we haven't, that we are looking for, is an ongoing project that I haven't tell you for the lack of time, but I can just mention that secretion of CYP in the A occurs by acetylation of the protein. So the so what happens is that you induce the acetylation of the protein is acetylated and released extracellularly, and then this acetylated protein has a role extracellular, and this is what is known for CYP. We know that parasite CYP is acetylated in epimastigotes. We didn't test in mastigotes, and
Yeah.
we
Yeah.
don't know the same mechanism, but whether the antibodies against the acetylated could be used. So we are now testing. We generate these acetylated peptides, and then we are going to test with this.
Yeah.
And
Sérgio,
then,
let let me put some two questions together because Julio is anxious to make his question also. Maria Cristina Mota and Vanderlei de Souza both asked about organelles, the whole of organelles in this process. Vanderlei asked about the peroxisomes of host cells that produces catalase. How can this catalase interfere with the CP in the cells? And Cristina would be suggest that it would be interesting to check oxygen consumption and ROS production during infection, and to see the whole of glycosome in this function.
Can you just repeat, because there was some interference? I couldn't. The Christina,
Yeah.
Mr. Christina.
Cristina suggested to check the whole of glycosomes if glycosome functioning could interfere with cyclphilin production.
Okay, I think they're just starting from this suggestion to see the metabolic state or how the glycosome respond to stress is interesting. Maybe I believe that the parasites can adapt to the metabolism to according to the level of ROS that
Yeah.
and this could change the differentiation
Yeah.
and certainly the metabolism could be driven by modifications of just the substrate, but also by modification of the enzyme. We now publish a paper showing that aldolase, once you
Yeah.
deactivate aldolase, you can increase the activity. So maybe under stress, you have acetylations controlling the metabolism. Regarding the Vanderlei question, but we have to examine. the glycosomes. The exosomes, whether the release...
The peroxisomes, the catalase present in peroxisomes with the role of catalase to broken hydrogen peroxide and
So
diminishing the
I
growth.
think this goes back to the cardiopathy and the myocarditis and things like that, because we started this work when we saw CPA. I show you this reviews about the story about the CPA in myocarditis, in inflammation and vascular permeability, and so whether this could be driven by the parasite if CPA19 is secreted. I think the importance of the story was to go through this, but we don't know whether exosomes will also play a role controlling that. I think it's a question of the pathology of the Chagas, or whether the parasite is able to sustain this environment or not.
Sérgio, let's hear Julio. Maybe he has some directioning of this conversation.
Okay, Julio.
Yes, okay. Sérgio, we have an interference.
Maybe your cell phone is too close. Yeah.
Okay. Now, Sérgio, can you listen to me?
Yes.
Okay.
Yeah.
Just exactly on that point that you were discussing now. I mean, in our model of angiogenesis, as I told you before, we have something very interesting in relation to your talk, because at the second stage, final stage of proliferation, probably close to the time of eclosion of trips, we have a booster of the angiogenic response. So we have a kinetic response in which it starts slowly, and it has a clear association with microvascular leakage in the early stages. And then there is a tremendous boosters, in 2 days. My question is related to, have you ever thought about bystander retention of what's the name? What's the name? Cyclphilin. Cyclphilin 19. Bystander absorption by cells that are neighboring cells, because I mean, when you have the burst of cells with a huge number of parasites, you would assume that you will have retention either by exosomes, as you mentioned, by the cells just in that surrounding. So since you have mentioned so many vascular effects, maybe the angiogenesis that we see as a booster, could be triggered by your enzyme. The second question has to do more with basic immunology, which is a very basic question, okay? Have you checked the level of MHC class 1 molecules in your myoblast infected cells when you have overexpression of cells? Because it would be interesting to know whether that could be part of a strategy of evasion, obviously, right? And the final question has to do with the amplification loop that we have once discussed. Have you ever attempted to inhibit with blockers of angiotensin II inhibitors? Because if you see an amplification loop involving the activation of this receptor, then it's interesting, even the role of ACE2, which now became so popular because ACE2, as you know, is a regulator of the activity of the angiotensin receptor signaling pathway, as well as bradykinin B1 receptor signaling pathway. So that could lead to a very interesting conjunction of research activities in your group, and also the stuff that we have been doing for a long time. And the model in vivo is very useful for that, because since you have now the knockouts and you have the wild type, we could easily try to evaluate whether the angiogenesis is actually compromised in this situation. And you have fluorescent parasites, so you have all the tools that you need in order to answer the question. Maybe you are wrong, but I think it's a good question.
Oh, just to answer all your questions, I'm really. I think you suggest very good experiments. I never thought of the MHC in the case of the smile to see if we affect. What we know for sure, and this has been shown by others, that these cells, if they cruzi infected cells, have a high level of ROS. So we confirm this data. The group of Luciano Andrade and also even Marcelo. So why nobody look for the MHC in this case is interesting. I don't know if somebody ever look at that. Regarding the release of the cyclphilin at the birth, I like this idea that when the parasites egress from the cell, many things are released, and we know that transaminase, and then it's not. I don't think it's only one molecule that is going to be responsible, but we can maybe test this and whether this is important in vivo. I think it's very. I will never try to do this. One of the problems to use the knockouts is that, remember I showed you that we still have 5% of the parasites with the cyclphilin. If you keep passing these guys in culture, we lose the knockout. This 5% take over. I don't know if this 5% is an heterozygote, is a new recombinant, something, and whether this is essential. I know there's a collaborator we are working on a paper together from Ohio State. He generated another strain, I think he used Brazil strain. He did the classical knockout by two steps, and he could not get the full knockout because there was an extra copy that reappears in the parasite. It just sequenced everything, and so he thinks the gene is somehow essential or in the long time. So we have to sort out this story to make good experiments. We tried to clone the knockout we have, and we got some clones, and then the clones were not clones. So we don't know if they reappear by recombination, if the clones don't grow. There's always this problem with the T. cruzi to get this full knockouts. So just to show that we still have some limitations on that. Regarding this final angiotensin, I like this idea that maybe there's a balance of the homeostasis and angiogenesis and many things once you infect the host with the parasites, and this is very important, and then we have to understand better this. And I'm sure that the things you do goes really together with this idea. We are just providing one extra protein. Maybe what I really try to do is whether this happens in patients, and then we look in humans, the chronic patients, whether they will have antibodies against the CYP-A. They don't have antibodies against CYP-A, and a few have against CYP-19. I don't know if there is any different type of lesion on this, if this is due to, and whether these antibodies are able to inhibit the function of the protein after all. So maybe the fact they are so similar makes the non-protective. So whether if you immunize a mice with CYP, and you can block something, so there's some experiments to do that. And we can even immunize with the N-terminus peptide. That is the one that makes the difference.
The difference, yeah. Sérgio, let's maybe turn to other cyclphilin. From YouTube, Marcia Paz is saying that your talk was excellent. and she suggested that you could take a look at the work that was just published by her group in BBA involving mitochondrial permeability transition pore and ROS and amastigogenesis. And at this point, at the same point, Emma Saavedra asked if CYP19 could affect the mitochondrial permeability transition pore of host cells. The opening of these pores would also be related to increased production of ROS.
Yeah.
And maybe Sérgio, would you say a word also about the method you used to measure ROS? It was a question here in our Meet chat.
Okay. So just say, Marcia, I look at paper, it's very nice. I see that.
Yeah, I was
And
sure.
I'm always concerned whether, because when you have the mitochondrial permeability transition pore is really involved in the ROS production in many systems, and cyclphilins are involved, and mainly cyclphilin D in the case. I'm sure that probably cyclphilin D participate in this process, whether this in the case of infected cells or not, or how this is modulated. We even tested whether I was curious to see if the only mechanism by which cyclphilin 19 induced the ROS would be through NOx. It could also affect the mitochondria because we are expressing that protein with some difference. So maybe this is happening in the cell. So I tried to measure the mitochondrial ROS. We couldn't do. We ordered the reagents to measure this mito-ROX. I'm not sure if the students we did correctly to see if these L6 cells have that. We also tried to see apoptosis, and what we know is there's no difference in the staurosporine induction of apoptosis, but we see a difference in nitrogen peroxide induction, but we don't know how to interpret exactly. So it's still, we have to understand more how that ROS is increased both in the cell and also perhaps in the parasite. Regarding the other question is how I measured that.
Yeah,
that image is using, it just in the microscope is not quantitative, it's just taking images at the same exposure with the cell rocks. And CFHTA is the most, we got the some advice, and this is considered one of the most appropriate measurements. We did that by adding the compound and measuring by fluorescence using a plate reader. We did, and also in solution after lysing the cells using a plate reader. I think it was on that.
Yeah.
We also normalized by the amount of cells using another stain. So we use this method.
And Sérgio,
indirect methods, yes.
We have a compliment on YouTube from Marcel Ramirez. Nice talk, Sérgio. Congratulations. I was also a postdoc in your lab many years ago. I would like to see if you did experiments to see the secretion of CYP19 during the cell cycle.
Yes, we haven't looked. One of the goals was to understand the secretion, and the key issue is they have these internals. I don't know if this has a role in the secretion or not. This is the first question. But and also CYP-A in mammalian cells is secreted. And I told you that secretion is due to acetylation of the proteins, and then this acetylation makes the protein migrate inside the endosomal compartment, and then is probably released by a kind of exocytosis. So it's not trapped in the, or it could be trapped in the vesicles. We published a paper showing the proteomic analysis of triple mastigotes, and we found CYP19 in the compartment, not membrane bound. So when we break the exosomes, we could detect CYP19 in the proteome. Whether this was an artifact like CYP goes together or is really secreted in these extracellular vesicles, we're not sure. We tried to spin down this supernatant by ultracentrifugation, and we could not spin down. So, but I think we have to do this more carefully to understand the secretion and whether the secretion occurs by modification of this of the acetylation, which could match with the mammalian's model. So whether acetylation could be a trigger of protein secretion or other modifications, or just is a cytosol that do this autophagic in multivesicular bodies as you work, and then or this is extravasicles bud from the space of the parasite. So it's still interesting to see how the secretion occurs. Thanks, Marcel. I still remember. We still have your things here.
Sérgio, we all thank you very much for your talk, for your answers, and it was very important to the students and to the cruzi community of our institute. We are very glad of could be here. Thank you very much.
Thank you all for attending, for your time, for the questions.
Yeah. Vanderlei would say some words.
No, only to thanks again, Sérgio, for this wonderful talk. I think that's opened a series of new ideas. Let's think about it.
Yeah, I think maybe cyclphilin could be useful to understand Chagas disease.
Yeah, well, did you check if the cytosolic cyclphilin are located in vesicles?
No, I think it's cytosolic completely.
Yeah, but because the association of actin is most useful for vesicle-associated things. If it moves in the direction of the cell surface to be released at some place.
It's important for the microfilaments movement. So all these chaperones, the cyclophilins, they have this role in sliding or moving them or assembling microfilaments, assembling or creating them.
Well, since you have it tagged with HA, I would suggest to look by EM to see where they are really located. The cruzi infected cells that we never characterize. Of course, some are exosomes released by a mastigotes inside the cells.
No, you're talking about this in the parasite. Okay.
In the host cells. I said the cyclphilin that you see a very intense labeling by confocal and super resolution microscopy in the cytoplasm of the host cells.
Okay. Yeah. Okay. And maybe somebody else is in. I think it's please.
Okay, sir. Thank you.
Thank you very much. Bye-bye.
Bye-bye.